Hello, good evening all.
Hello, good evening.
Am I audible? Can anyone unmute themselves and let me know whether I'm audible or not? Okay. Okay, so I'll start sharing my screen and just let me know whether you are able to see my slides or not. Just are my slides visible? Okay. Okay, so good evening all. So I welcome you all in our first problem solving session for week 1 for the course Genome Editing and Engineering. So this course is being taken by Professor Utpal Vora. He is professor at Department of Biosciences and Bioengineering at IIT Guwahati. So I am one of the TA for this course. So we will be having problem solving sessions where you will learn some basic concept of for the course, and this will help you to solve your own assignments which might have already be given to you for the week 1. So today the lectures we will be covering, the questions we will be covering are from lecture 1, which involves gene and genome organization, lecture 2 history and basics of genetic engineering, and lecture 3 which includes advantages and limitation of genetic engineering. So I briefly introduce myself as well. So I am Pooja Yadav. I am a PMRF fellow and a PhD scholar at Department of Biosciences and Bioengineering at IIT Bombay. I am a PhD student in molecular virology lab. So so the most of the lectures for me will be we will be having questions, and for each questions I first like you to try to answer for each of the question, and then we will look into the concept for each of the question. Just wait a second, people are still joining in. Okay. So yeah, as I told, we will be having questions. We will try to solve each of the question, but first I am expecting you to try to answer the question first, then we will look into the concept for each of the question. So shall we begin with our lecture, or do you guys have any query or something? I hope I am audible to everyone. Okay, so let's go to our first question. Um, so the first question is who coined the term genome? So you can go through the options and try to answer this question. Okay, so we in chat box we have two options. One is C and one is D. Are there any other option which might be true apart from C and D? Okay, so most of the people have answered C. Few of have answered D. Okay. Are there any other option that might be correct apart from C and D, which you think might be the correct answer? Guys, please try to join by 6:10. Okay, so the correct option for this question is option D, Hans Winkler. So let's see for each of uh, them who discovered what are their contribution in the field. So the first option is Frederick Griffith. So he was mainly behind the Griffith experiment, which we all might be knowing, where he discovered the phenomenon of the transformation in bacteria. So what he did, he was working with the Streptococcus pneumoniae. He was trying to develop vaccine for the Streptococcus pneumoniae. While working with the Streptococcus, he found uh, that the non-virulent bacteria could become virulent by absorbing genetic material from the dead virulent bacteria. So while he was doing his experiment, so he had basically 4 conditions in his experiment. One is the live S or the Streptococcus strain, which is virulent, and the other one is live R strain of the Streptococcus. So the S strain is basically the smooth form of the Streptococcus, while the R has the rough outer covering or the off for the Streptococcus. So the S strain was virulent, but while the R strain of the Streptococcus was non-virulent. What he saw while he injected this virulent strain into the mouse, the mouse died. Of course, it would die. But when he injected the R strain, which was non-virulent, the mouse lives, which is again uh, uh, straightforward that mouse would live in the non-virulent infection via the non-virulent bacteria. But when he heat killed the S strain and injected into the mouse lives again. If he is if we inject the dead bacteria into something, it won't cause the infection. But what he saw, when he mixed the live R strain, which was non-virulent, with the S strain, which was virulent, the dead S strain, and then injected into the mice, then the mouse died. But here we can see that the S strain that he used was dead, and the non-virulent strain was alive. But he saw that uh, even the dead strain was used for the virulent strain, the mouse still died. So then he concluded that something is being transformed into the S strain into the R strain by the S strain. There is something which is being transferred from the S strain into the R strain and making it into the virulent one. So he showed that the non-virulent bacteria could become virulent by absorbing genetic material. So he did not know that it was a genetic material. What was the material that was being transferred? He just concluded that something was transferring or transforming the R strain into the virulent one from the dead S strain. So further, uh, from different scientists, they identify it was a DNA that was being transferred from S strain to R strain. So that was his contribution. The second option was Francis Crick. So along with the Watson, he discovered the structure of DNA. So they proposed that DNA is the double helix double helix structure, and further he also contributed in the genetic code, which we will see later on, and the central dogma, which is basically how DNA replicates itself and then transcribe into RNA and then translate into a protein. So that was his discovery. And Just a second. Uh, so those who join late, please try to join max by 6:10, so we don't have to interrupt the session in between in order to admit you guys. So it's a request if you join by 6:10. Okay, sorry about that. So the third option, so this guy, this he coined the term gene, genotype and phenotype. He also showed the difference between gene, genotype and phenotype. What is the gene? So gene is basically the segment of DNA which so we have a very long DNA strand, but in eukaryotes the entire DNA does not code for protein. There is only a certain region in the DNA which codes for a protein. So this region is known as gene. So, and the genotype is the entire making of the genetic material, uh, including the DNA sequence that determine inherited traits. And the phenotype is the set of observable traits or characteristics we see, like the eye color or the hair color or our height. So these are the phenotype, and these are the factors which determine these phenotype is our genotype. So this was his contribution. Further, Hans Winkler, so he was the guy that coined the term genome. So what he defined was the genome as a complete set of chromosomes. In human, we have 23 pair of chromosome, 22 pair of chromosome, while one is again X and Y apart from those 22. So the entire set of chromosome, it is termed as the genome. So this was coined by Hans Winkler. So the correct option for this question is option D. So I hope this question is clarified, or do you guys have any doubt in this question? Okay, so if it is fine, so let's move to our next question, question number 2. For the 64 codons, which of the following statements are true? The first option is 61 codon for amino acid, while the remaining 3 are stop codons. B option is 60 codons code for amino acid, the remaining 4 are stop codons. So you can go through the option and try to answer. Okay, so we already have answer in our chat box. So okay, most of the people are giving answer A. So is there any other option that might be correct apart from option A? Okay, so everybody is giving the answer A. So the correct option for this question is A only. So 61. So we have 64 codons. So we have 4 nucleotide acid and 20 amino acid. And the combination, the 3 genetic code or the codon, they code for 1 amino acid. And from the combination of these 4 nucleotides, we have 64 total codons, out of which 61 codes for amino acid, different amino acids, while the 3 are the stop codons. They don't code for any amino acid. They stop the translation process of a gene. So what is a genetic code? Genetic code is a set of rules defining how the 4 code of DNA, like ATGC, is translated into 20 letters of amino acid. So if you think we just have uh, uh, four nucleotides, four A, T, G, C, but we have 20 amino acids. So how these 20 amino acids are being coded by just four nucleotides or base pairs? So later it was found that the combination of three base pair codes for one amino acid, and which are the building block of the protein. So the genetic code is a set of three-letter combination of nucleotides, which are called codons, each of which corresponds to specific amino acid or stop signal. So many of them codes for amino acids. Uh, the different codes they can code for the same amino acid, and apart from the apart from the codons which codes for amino acid, there are three codons which does not code for any amino acid, so they are called stop codons. So uh, the Marshall and Heinrich, they performed experiment for deciphering these genetic code, and they synthesize a synthetic RNA which had just U, and they found that this U was coding for phenylalanine. So later, different scientists, including Hargobind Khurana, they identify the rest of the amino acid that was being transcribed from different genetic code. So the correct option for this is option A, 61 codons codes for amino acid, while the remaining 3 are the stop codons. So I hope this question is clear. Okay, if this is clear, let's move to our next question. Question 3. The question 3 states, histone DNA interaction are mediated by what type of forces? A is water mediated hydrogen bonds, B is ionic interaction, C non-polar interaction, D all of the above. We have two options, B and D, in the chat box. Is there any other option that might be correct apart from B and D? Okay, the correct option for this is option D, all of the above. So the main interaction between the histone and the DNA is by the hydrogen bond, ionic interaction, and non-polar interaction. So this, if we consider human DNA, the human DNA is basically around 2 meters long. If we uh, if we try to calculate the entire length of the human DNA, it is around 2 meter long. But you can see that this entire 2 meter long DNA is compacted in a nanometer nucleus of our cell. So how this 2 meter long DNA is present in a nucleus which is itself in nanometer range? So how it is possible? So it is possible because of the DNA is packaged in such a way that it can go and the entire length, the entire 2 meter long DNA can go inside the nucleus. So how this DNA packaging occur and what are the main forces, what are the main interaction which leads to such packaging? Like we can see, like I'll share this video. So where you can see how this packaging of the DNA occur. So I'll just briefly stop sharing the screen and again reshare the screen if it is fine. Just wait for a second, I'm having some issue. Uh, can you hear is the video audible to you guys? Can you confirm? Okay. Okay, I'll play the video. I hope the video was clear. So as you saw, like how the DNA compaction in the nucleus occur with the help of the histone protein, and the interaction which, and like the bonds or the forces which are involved in the interaction of the histone and the DNA are the hydrogen bonding, the ionic interaction, as well as the non-polar interaction. So this, as you saw that we have our DNA double helix, and how the the DNA is coiled around the histone protein, uh, uh, which further this coil, this the coiling of these histone protein along with the DNA, they forms the nucleosome, which is around the 10 nanometer in the diameter, which further supercoils into 300 nanometer which further supercoiled in 30 nanometer diameter, which is the chromatin, which further supercoils in 3000 nanometer diameter, and further we have our chromosome, which is around 700 nanometer. So as you can see, like how this compaction has occurred, like in this, the strong compaction of DNA around the histone protein has occurred. So the interactions, the forces are the ionic interaction. So we know that DNA is negatively charged due to its phosphate backbone, and these histone protein, they are positively charged, have the positively charged amino acid like lysine and arginine. So there is, because of these opposite charges, it leads to the strong electrostatic or the ionic attractions, uh, which are the primary force in for the DNA and the histone interaction. And the second was the water mediated hydrogen bonding. So the water molecule can mediate hydrogen bonding, we know, between the histone and the histone proteins and DNA. And the third one was the non-polar or the hydrophobic interaction. So these are less prominent than the ionic interaction and the hydrogen bonding. So these hydrophobic interaction between the non-polar amino acid chains of the histone protein and the deoxyribose backbone of the DNA, or between the histone surface also, all these interactions, they contribute to this nucleosome stability and also in the compaction of the DNA along with the histone. So the correct option for this question is all of the above. So I hope this question is clear. Okay, so if it is clear, let's move to our next question. Question number 4. The question number 4 states the first type 2 restriction enzyme to be identified was, first is BAMH1, second is EcoR1, third is HIM3, fourth is TAT2. So C option is HIM2. So we have C as our answer in chat box. So is there any other option which might be correct for this question apart from option C? Is there any other option which might be correct from apart from option C? So for this question, the correct option is C, into. So let's see. So first we'll see what are the restriction enzymes, and why are we even talking about restriction enzymes. So restriction enzymes are basically a protein or enzyme, you can say, which cuts the DNA at specific location. So there are two sort of restriction enzymes, which are exonuclease, which cut at the terminal of the DNA, either at the 3' end or of the 5' end of the DNA, and the other one is the endonucleases, which can cut the within the DNA or the RNA molecule. So why are we talking about the restriction enzyme? Because in the genetic engineering, so when we are working with our gene of interest, so when we make the recombinant plasmid with our gene of interest, when we want to study the phenotype, and when we want to study the protein which are being translated by our gene of interest, and we further want to study on the protein of our interest, what we do is we take the gene from the genome of the organism, or it can be from anything, from a virus, bacteria, or plant, or any human cell. We take the gene, we amplify the gene with the help of PCR, and we combine the gene with our plasmid of interest. So what we do is we cut the gene as well as the plasmid of our interest with the same restriction enzyme. So it's sort of, they create a blunt end, which here you can see this, we have cut the sequence with EcoR1 enzyme, and it has sort of created a blunt end. This part of the single stranded part of the DNA is complementary to the to this part of the DNA. So when we cut our plasmid as well as our gene with the same enzyme, we get these sort of complementary sequences, and when we join, so further we can join our gene of interest with the plasmid which we are working with the help of these restriction enzyme. Because they have the same, we are using the same restriction enzyme, they will go and cut at the same site, and they will produce these overhanging sequences which are complementary to each other. So later on they can go and join, the gene can go and join to the plasmid. And there are two type of restriction enzyme, which is exonuclease and one is endonuclease. So these exonuclease, they cut at the terminal of the DNA, and the endonuclease, they cut within the DNA. So we basically use the endonucleases because the gene, because the plasmid we use is basically circular plasmid generally we use. So we need the endonucleases because it has to go and cut within the DNA sequence, not because there is no terminal end in the circular DNA. So the commonly we use endonucleases, and in the endonucleases again there are certain type, which are, first is type 1. So these are differentiated via the cleavage site, where they actually cleaves, or location of methylase. So type 1 restriction enzyme are those which recognize a certain site, but it cuts at around 100 base pair away from the restriction site. And the type 2 is the restriction enzyme which actually cleaves at the restriction site, while the type 3 restriction enzyme is again a restriction enzyme which cleaves 24 to 26 base pair downstream the restriction sites. So what, for our purpose, we need a restriction enzyme which can cleave at a certain location, at a specific location, not at a very random place. So that is why in the genetic engineering, the type 2 restriction endonucleases are mainly used. For example, EcoR1, BAM-H1 and HIND3. So these are commonly used restriction enzyme in when we work with genome engineering. So the first restriction enzyme that was discovered was HIND2. So it was discovered by Hamilton. So later on, he also also received Nobel Prize for his discovery, and the site which is recognized by this enzyme. Just a second. Is AAGCTT, and it cuts at this between this AA base pair. So the correct answer for this question is HIND2. So I hope this question is clear. Okay, if it is clear, let's move to our next question. Question 5 states, the DNA from which two organisms did Paul Berg use to create the first recombinant DNA? Please go through the option and try to answer this question. We have two options in the chat box, option B and C. Is there any other option which is correct, which might be correct apart from B and C? Okay, so the correct option for this question is option B, SV40 virus and lambda phage. Just a second. Those who are joining in late, please try to join max by 6:10 from the next class. So else we need to interrupt the session and then I need to come back. So please, it's a request, try to join by 6:10. Okay, so let's continue with our session. So the correct option for this question is option B, SV40 and lambda phage virus. So this Paul Berg, he created the first recombinant DNA. So this uh, the recombinant DNA is again comes under the genetic engineering, where we modify the genome of our interest. Again, the genetic engineering is a process that uses the laboratory based technologies to alter the DNA makeup of an organism. Or if we want to study our gene of interest, we can take the specific gene and further clone into our vector. So this may involve the changing a single base pair, deleting a region of a DNA, or adding a new segment of the DNA, or just to simply study the gene function, where we don't change the gene makeup and the gene has not been studied previously, and we want to study the this gene, and it is coding for some hypothetical protein or any other specific protein which had not been studied in the past. So we can take that particular gene and study it. So how do we do it? So the commonly we have our plasmid DNA. So plasmid is basically the extra chromosomal DNA which is commonly present in the bacteria. So bacteria has the main DNA and the extra chromosomal DNA. So this we can isolate this plasmid, and we can modify this. We can either isolate this plasmid, and we can also insert this plasmid in the bacteria. So in from the past there has many modification in the plasmid, where we can add the plasmid basically should have three main sites. One is the origin of replication, where the replication starts. Any selection marker gene, with the help of which we can select our true recombinant DNA or the plasmid, which can include the antibiotic resistance gene or some other laxy gene. And we also have some sites where we can actually insert our clone our specific gene. So we need to have some restriction site. So if we want to clone, so let's suppose this is our plasmid, and if we want to clone our gene of interest at this site, so here we will be having one restriction in site, and here we will be having different restriction site. So to make sure that these restriction sites should not present in our specific gene, like inside the specific gene, because if it will be present, the restriction enzyme that will be used, it will go cut our DNA or our gene of interest into multiple fragments. So make sure that this restriction enzyme you are not using, the site for these restriction enzymes should not be present inside our gene of interest. So when we amplify our gene of interest, what we do is we insert these restriction site into the primer. So, and these restriction site are present at the terminal of the gene, not inside the gene, and we add these restriction site from our side. They are not naturally present. So then we cut the plasmid as well as the gene of interest with the same restriction enzyme, which again will have the complementary overhanging sequence in the plasmid as well as in the gene of interest, and then it will get ligated with the help of enzyme ligase, and then we transform this the recombinant DNA again into the bacteria, and then we can amplify this plasmid or the recombinant DNA. And if you want to study the protein which are being expressed by this gene, we can also get the protein from the bacteria. So genetic engineering, it may involve adding a gene from one species to an organism from a different species to produce a desired trait. It has a wide application in research and industry. It is production of cancer therapies, brewing yeast, genetically modified plant, livestock, etc. So the first recombinant DNA that was created was by Paul Berg. He created the first recombinant DNA by combining the DNA monkey virus SV40 with that of the lambda virus. So it is the DNA of the SV40 virus and it is the lambda phage DNA. So what he did is so the it uh, these two DNA, they were linearized by the restriction enzyme EcoR1, and again these exonucleases, the lambda exonucleases, they were used to remove the base pair from the 5' end of the DNA uh, to increase the ability. So he used a slightly different approach because he was doing for the first time. Now we know the easier method. Now we just add the two restriction enzyme that will create a overhanging sequence. But imagine in a very recent year in 1972 when the guy was first trying to recombine these DNA. So what he did to get these overhanging sequence, now we use the endonucleases, right? So first he used one endonuclease, just one EcoR1. It cut the DNA at the specific restriction site. Then he used an exonuclease which cuts at the 5' end of the DNA. Uh, my screen sharing is disabled. Okay. So it was like that from earlier, or you could see the screen from past 5 minutes. Okay. Now you can see the screen. Okay. So please just let me know as you are not able to see the screen, because I am always sharing my screen. So if you are not able to see it, just let me know immediately. So where I was talking about the principle of genetic engineering. So how we clone our gene of interest into the plasmid. We have two restriction sites in the plasmid, as well as we add the restriction site in our gene of interest. Then we cut the plasmid and the gene of interest, and then we combine, because we'll get the overhanging complementary sequence because we use the same restriction enzyme. Then we combine the plasmid and the DNA and the gene of interest, and then transform into a bacteria or other cells to get our desired recombinant plasmid. Is my screen still visible? Slides? Okay. I think something is, I'm not sure why. Okay. Okay, so as I was telling, so this is using the same restriction enzyme. We can add our gene of interest into the plasmid, and then we can study our specific the trait of the gene or the protein which are being coded by this particular gene, or we can use these protein in industry application, like as we know the insulin is being translated by this recombinant plasmid only. So the first recombinant DNA that was created was by Paul Berg. He combined the two DNA from two viruses. One was monkey virus SV40 and the other one is lambda virus. So he used some sort of different technology. The principle was similar, but he was doing his experiment. He was the first one who was doing the experiment and tried to do that. So what he did was, first he used the endonuclease, which could go and cut between the DNA sequence, and further he used this exonuclease. So exonuclease, what exonuclease would do, he used the lambda exonuclease, which cut at the 5 prime end the single strand of the DNA. So because it was cutting at the 5, just only the 5 prime end, so it now we it now we have the overhanging sequence at the 3 prime end in both the monkey virus as well as the lambda virus. So then, because now these are the complementary sequence, these are the overhanging complementary sequence, then he later on joined these sequences with the help of the terminal transferase. So this is how he recombined the monkey virus SV40 with the lambda virus. He was the first one to create the recombinant DNA, and this is how he created. So I'll share 2 articles with you. So if you want to read more about his work, so you can also read. So these are interesting paper and a good article. So if you have time, so you can read about it. So I've shared the link in the chat box if you guys have time and if you are interested, so you can read about it. So the correct option for this question is option B, SV40 virus and lambda phage virus. So I hope this question is clear. Okay, if it is clear, let's move to our next question. Question 6. Question 6 states, tumor-inducing TI plasmids are derived from which organism? So please go through the answer, go through the option and try to answer this question. We have option C as our answer in chat box. Is there any other option that might be correct for this question apart from option C? I think my screen sharing stopped again. I am not sure why it is happening, so please bear with me, guys. So the correct option for this question is option C, agrobacterium tumefaciens. So this TI plasmid, these are the plasmids that are derived from the soil bacterium, Agrobacterium tumefaciens. So these bacteria are known for their ability to cause a disease which is crown gall disease in plants by transferring a segment of its DNA, which is called the tDNA. So this is the plasmid, this is the Ti plasmid, which is present in the bacteria, Agrobacterium tumefaciens. So here you can see this region, which includes these sequence. This is called the tDNA region, and it is responsible for causing a disease which is crown gall disease, which are like tumor sort of thing on the plant into the plant genome. So what they do, what happens is this region it goes and combine with the plant genome, and then there is the no regulation of the replication. There is the hindrance in the regulation of the replication process of the plant genome, and this uncontrolled cell division occur, and which leads to the formation of the tumor for the plant. So it leads to the uncontrolled cell growth and the tumor formation. So the Ti plasmid, or the tumor inducing plasmid from the Agrobacterium tumefaciens, they have a major role in a plant genetic engineering. Why? Because again, so as I've told you, this region of this plasmid has the ability to go and insert itself into the plant genome. So it goes and get incorporated or integrates into the plant genome. So in nature, the Agrobacterium transfer a portion of this Ti plasmid, this is called the tDNA, into the plant cell, where it integrates into the plant genome. So what now the scientists are doing? So they are replacing the tumor causing gene, or the tDNA, with their gene of interest. So if you swap this region with your gene of interest, this plasmid is still able to integrate this part into the plant genome. So now the scientists are inserting their gene of interest for it might be for plant growth or for some other phenotype. They are inserting the gene of interest and then infecting the plant with the virus which is containing this plasmid with the bacteria which is containing this plasmid, and this plasmid this bacteria transfer this part of the plasmid into the plant genome. So we have the Agrobacterium tumefaciens, then we isolate the tDNA plasmid, then we have our Ti plasmid. Again, the basic process is same. We cut the plasmid using the restriction enzyme, and we also cut our gene of interest with again the same restriction enzyme. Then we integrate our gene of interest into this plasmid. This forms a recombinant plasmid which has our desired gene. Then this Ti plasmid is again transformed into the bacteria, again the same bacteria, Agrobacterium, and this tDNA carrying inserted gene incorporated into the plant cell chromosome. Then we infect this bacteria into the plant, and where our gene of interest goes and incorporated into the genome of the plant. Then we have a plant which has the phenotype of our interest, because we have inserted our gene of interest into the plant. So we might produce a plant with the phenotype we desire. So this is how these plasmid from Agrobacterium tumefaciens are used for genetic engineering purpose. I think my screen sharing has stopped again. Please bear with this, guys. I will figure it out from the next class. For the patience. So the correct option for this question is option C, Agrobacterium tumefaciens. So I hope this question is clear. So if the question is clear, let's move to our next question, question 7, which states that which enzyme catalyzes the formation of a phosphodiester bond at a single strand break. First option is phosphodiesterase, B is DNA ligase, C is polynucleotide kinase, D is alkaline phosphatase. So please try to answer this question. We have two options, option B and option D. Is there any other option that might be correct apart from option B and D for this question? Okay, so the correct option for this question is B, DNA ligase. So let's look into the concept for this. Okay, so when we insert or when we cut DNA with the endonuclease, we know that we get an overhanging complementary sequence when we use the type 2 endonucleases. It cuts at the specific restriction site and we get the overhanging this complementary sequence. And but for them to ligate or for them to join at the correct location, we need one enzyme which is the DNA ligase. So here you can see we have cleaved our there is the cleavage at a particular restriction site, and it will lead to the formation of the overhanging sequence, the complementary sequence, which we call sticky ends. Okay, so there are certain enzymes which does not lead to formation of these complementary sequence. So here we see that this enzyme has cut at the extreme region of the restriction site. So this leads to the sticky end, but there are some enzyme which cut at the middle of the sequence, which leads to the blunt end as well. So if we want to stick these sticky ends as well as the blunt end together, we use the DNA ligase enzyme. So what the ligase enzyme does is, so now after the restriction, we have this phosphate group on the 5' end of the DNA, while this OH group at the 3' end of the DNA. Okay, my sharing has again stopped. Again, sorry about that. So this DNA ligase, it helps in the formation of the phosphodiester bond between this 3 prime OH group as well as the 5 prime phosphate group, because when we cut the DNA we'll get one 3 prime end as well as the other 5 prime end. So this T4 DNA ligase, it helps in the ligation and the formation of the phosphodiester bond between the two nucleotides which are at these locations. So the correct option for this question is DNA ligase. I hope this question is clear. Okay, if it is clear, let's move to our next question. Question number 8. The genome of a lambda phage consists of: first is double-stranded linear DNA, B is single-stranded linear DNA, C is single-stranded linear RNA, D is double-stranded circular DNA. Please try to answer this question. We have two options in the chat box, option A and B. Are there any other options which might be true apart from option B and C, A, B and C? The correct option for this question is option A, double-stranded linear DNA. And why the lambda phage is important? Because this phage is also used for the genetic engineering to transfer our gene of interest. So I have a video to show how these lambda phages are being used in the genetic engineering of our particular gene. So I'll briefly stop sharing the screen and again reshare with the video. I hope you guys are able to see the screen. Can you hear the audio? Okay, so I'll start. So the lambda fast DNA is used as a vector to where we can insert our specific gene, or then we can infect the this lambda phage to the bacteria where the gene of interest can go and get integrated into the bacterial gene, so we can have our desired gene inside the bacteria. So these lambda phage, they can act as a vector. So I hope this question is clear. Is the question is clear, or you guys have any doubt in the question? Okay, if it is clear, let's move to our next question. Question 9. Who coined the term replication fork? Please go through the option and try to answer this question. So we have two options in the chat box, option A and option D. Is there any other correct option apart from option A and option D? Okay, so the correct option for this question is option A, John Keynes. So, replication fork is basically during the DNA replication, while the firstly the two DNA, so we know that DNA is double helix, so the two strands has to be separated for its replication. So when the two strands they get separated, they form this Y-shaped structure, and the point where this Y shape these two interacts, this is the replication fork. So at this point the replication starts. So let's see how the replication of the DNA occur in this video. So I'll again briefly stop sharing my screen and I'll share the screen again. I hope my screen is visible and you can hear the audio as well. I hope the process of replication is clear, and the term the replication fork was coined by John Keynes. So I hope this question is clear. Okay, so if it is clear, let's move to our next question. Uh, the uh, the flavor served on tomato has an increased shelf life due to suppression of which gene? So please go through the option and try to answer this question. We have two options, option A and option D in chat box. I'm not sure why people are still joining in. So, is there any other correct option apart from option A and D, which might be correct? The correct option for this question is option D, polygalacturonase gene. So let's discuss. So the flavor saver tomato, it was the first commercially grown genetically modified whole food crop. So before that we had genetically modified cotton, but we did not have the whole food crop. It was the first commercially genetically modified whole food crop which was modified to ripen without the softening. So initially what happened was the tomato used to for when it used to get ripened, it also used to get very soft, which then becomes very hard to maintain and to sell and to eat. So there was very loss in the market because of the softening of the tomato. So this how they actually make this tomato, which uh, now get ripened without the softening. So this softening of the fruit is basically largely due to degradation of their cell wall, which is made up of pectin by an enzyme which is polygalacturonase and PG enzyme. This gene encode PG, it has been isolated and cloned. So as we know that our DNA has two strands. One is sense and the other one. So the one we call is sense strand. The other one which is complementary to it we call it as antisense strand. So only and now we have the transcription process where we get our mRNA from the DNA. So uh, uh, the sense uh, the mRNA will code for a protein only when it is when we get the mRNA from the antisense strand, because this antisense strand would lead to the formation of the sense mRNA because it is complementary. So only those mRNA will code for a protein which is sense or the positive in nature, not in the negative in nature. So what scientists has done, they have isolated the gene for first they have isolation of the DNA from the tomato plant which codes for this enzyme polygalactic or the PG enzyme. Then they transfer the PG gene to a vector and the production of the complementary DNA molecule. So in the normal plants, this PG gene encodes for a normal or sense mRNA that produce the enzyme PG, and it is actively involved in the fruit ripening. After the cDNA of the PG, it encodes the antisense mRNA, which is complementary to the sense mRNA. So during the cDNA, what we do is first we have the RNA. So we have the DNA for this PG gene. Now we have we now it we have the mRNA because this gene has been transcribed into mRNA. Then we isolate the mRNA from the plant and we make the complementary cDNA. So this complementary cDNA will have now the antisense mRNA, because in cDNA complementary DNA synthesis, what we do is we synthesize DNA from the mRNA. So now after this complementary DNA synthesis, we have the antisense mRNA. So now we take this antisense mRNA and we hybridize between this sense and the antisense mRNA. So when we hybridize this sense and the antisense mRNA, this mRNA won't lead for the production of this protein. So this is how they use the genetic engineering to modify the tomato, where there is no production of this PG enzyme, which then there is hindrance or inhibition of the softening of the or ripened mango. So this is how they stop the protein sequence of this PG, and this lead to have this resulted in a tomato which gets ripened, but it does not get softened very easily when it ripens. So this is how they had make this use the genetic engineering to modify this tomato. So the correct option for this question is option D, polygalacturinase gene. So I hope this question is clear. So if you have any doubt, you can ask. I think my screen is so this was our last question for this lecture, for week 1 lecture. If you guys have any question, you guys can ask if you guys have any question from your side, you guys can ask, because this was our last question for this lecture. If you guys don't have any questions, so you guys can leave the meeting. So there is one announcement. So I will be having lectures on each Tuesday at the same time. So we will be having the next lecture on Tuesday at the same time for discussing the week 2 lectures. So if you guys don't have any doubt, you guys can leave the meeting, or else you can ask any doubt right now. Uh, I will not share. I will upload the PPT, and there will also be a link of this recording as well. So I think you guys will get the slides as well as the recording, most likely. Okay, so I'll stop sharing the screen. Thank you all for joining in for today's lecture.